In this second video here, we're going to talk about salmonellosis, or the disease caused by salmonella. You've all heard of salmonella before. There was just an outbreak of salmonella associated with red onions, and those diseases are caused by a single species of bacteria, Salmonella enterica. What's interesting about this species is that there are a number of what are called serotypes. This is just a different word that means strains, different subtypes of bacteria within that species. They get shortened to something like Salmonella typhi or Salmonella paratyphi, but the true name would be Salmonella enterica serovar typhi, which is a really long name, and hence we shorten it to just S. typhi. These two serotypes, typhi and paratyphi, are responsible for the disease typhoid fever, which is a very severe form of salmonellosis. The more common types that we see here in the US are Salmonella enterica serovar enteritidis and serovar typhimurium. Typhimurium is actually what we've used in lab, and these, again, they cause salmonellosis in the US, salmonellosis being the less severe form of the disease as compared to typhoid fever. So Salmonella enterica, Salmonella species are motile and they have peritrichous flagella, which means the flagella are found all over the cell body, like so. And they are, again, gram-negative bacilli, just like Shigella, just like E. coli. There are more than 2,000 different strains or serotypes of Salmonella enterica, which is a part of why it's so hard to create a vaccine against bacteria such as this. And of course, you guys are aware of how salmonella is acquired. Most often we hear about eating raw eggs or undercooked chicken, but as we know with the current or the just resolved outbreak, it can often come from produce as well. Red onions in this latest one. There have been outbreaks associated with cantaloupe and other fresh fruits and vegetables. But one thing people often don't know is that reptiles are a really common source of salmonella infection, particularly turtles. Reptiles have co-evolved with salmonella so that it's part of their normal microbiome, just like we have co-evolved with E. coli. So it's a really interesting evolutionary relationship between the bacteria and reptiles. One of the reasons salmonellosis can occur so often and outbreaks can happen is because it's a very, very low infectious dose. That means the amount of bacteria necessary to set up an infection. And for salmonella, that's anywhere between 1,000 and 10,000 cells. That sounds like a lot, but recall that a single colony on a plate can have thousands and thousands of cells. And we compare salmonella with its 1,000 to 10,000 to something like cholera, which actually requires about 1 million bacteria to be ingested in order to cause disease. So this is a pretty low infectious dose. Following the process of infection here, salmonella attaches to the epithelial cells lining, in this case, the small intestine. So Shigella was in the colon, Salmonella is in the small intestine. Similarly, though, it does trigger endocytosis and get into the cytoplasm of our cell, and it replicates inside that food vesicle. Okay, so it does not get killed, it does not get destroyed, it actually replicates there in our vesicle. From here, salmonella does kill the host cell, and this results in inflammation like fever, intestinal cramps, and diarrhea, the main hallmark of salmonella disease. And as these cells die, the bacteria can make their way into the bloodstream, like we saw with Shigella. The difference here is that bacteremia occurs. That means we do see salmonella in the bloodstream. And this can be a diagnostic difference between salmonella and Shigella. Similar to pretty much every other diarrheal disease, salmonellosis results in a high fever. As the intestinal cells are dying, it causes inflammation that causes fever through the whole body, abdominal cramps, and again, bacteremia or bacteria in the bloodstream. And this is very, very different from our other diseases. In very rare, very severe cases, salmonella infection can lead to bowel perforation. As those epithelial cells die, a perforation or a hole in the small intestine can occur, and salmonella can leak out into the peritoneal space and cause a very, very severe infection. Fortunately, this does not happen very often. We can diagnose it using that same X-tag test that we used for Shigella, and in a similar vein, usually salmonellosis is self-limiting, so we'll send our patient home, tell them to drink lots of fluids, replace electrolytes, otherwise rest, and the disease will clear in a few days' time. If it's very severe, we see bacteremia, our patient is experiencing a bowel perforation, or if our patient has plain old typhoid fever, we're going to give them an antibiotic because those can be life-threatening situations. But otherwise, it's self-limiting. We tell our patient to look out for themselves and come back if they get worse.